I'd like to thank the organizers for the opportunity to present in this Exhibitor Spotlight webinar series. My name is Sheryl Denker, and I'm going to present to you today in collaboration with my colleague Alistair King the ways in which working with Eurofins discovery can improve the chance of success of oncology drug discovery programs, especially with relation to early life cycle solutions. We are all aware of the challenges of drug discovery, and this is not really unique to oncology, but in fact a common theme that is seen across all therapeutic areas. There is a tremendous increase in the expense of drug discovery, especially over the last 10 years, and overall processes used are still relatively inefficient from early stage R&D all the way through to preclinical candidate selection. Importantly and unfortunately, many preclinical models that are actually used have relatively poor relevance to human disease biology. Ways in which we can reduce risk and improve efficiency in the drug discovery process hopefully can drive down some of the expenses. But more importantly even, we need to improve the chances of success of new anti-cancer agents being developed for patients who are very much in need. One way of stepping up to this opportunity, then, is to work with a CRO such as Eurofins Discovery with a comprehensive and diverse portfolio of capabilities that are directly relevant and suitable for use in oncology drug discovery. For example, we have a large number of state-of-the-art pieces of equipment and equipment platforms that can be used to help drive oncology solutions. We have a number of specialty platforms. As example here are listed just a few, such as our KinomeScan, Biomap, and OncoPanel platforms, all of which can be used in a variety of ways to suit different oncology drug discovery programs. These niche or unique human-centric platforms can help distinguish your preclinical discovery and thus enable better decisions to be made as one progresses from early discovery through hit finding to lead optimization and ultimately to the selection of a preclinical candidate for eventual clinical evaluation. We have a number of approaches that are highly translationally relevant. This improves the chances of success in the clinic. I'll talk a little bit more about that in subsequent slides. In addition to the technology platforms, we have tremendous scientific and regulatory experience among the staff at Eurofins Discovery, and we can bring any aspect of this to bear on an oncology drug discovery program to help reduce inefficiencies, reduce risk, and improve chance of success. We have committed staffing and global operations across a range of sites and engage our team not only to improve drug discovery programs, but also to help uphold things like client IP and program confidentiality. In addition to small molecule drug discovery, which has long been the mainstay of oncology drug discovery programs, we also support programs that are using biologics or a wide range of biologic therapeutic agents. This is an emerging theme in more recent years, and we'll talk a little bit more about that in subsequent slides. In terms of oncology drugs, looking at 2020 from a regulatory snapshot, on the left we can see that both for the EMA and the FDA, more than half of the drugs approved in 2020 were actually anti-cancer agents, either as direct anti-cancer or immuno-oncology therapies. Of those two different approaches, a significant number were actually newly described active substances. When one looks a little more closely at those drugs that have been approved on the right panel, we can see that immunotherapies are still a growing area of therapeutic modality. This has been driven in part by the advent of immuno-oncology agents in particular, but also by the expansion of ways of looking at targets that have conventionally been suitable for immunotherapy using a variety of biologics as therapeutics. Chemotherapies and small molecules, however, still constitute a significant portion of these approaches and shouldn't be overlooked. We're seeing approximately the same number of small molecule agents approved by class as the immunotherapy agents. Ultimately, then, when one thinks about the agents being approved, a variety of different modalities are important and, in fact, in some cases can be combined, such as we see with antibody drug conjugates. When we look at disease incidence, one of the consequences of the COVID-19 pandemic that has gripped the world for more than a year and a half now, we really worry about the potential increase in cases that are seen for a variety of different cancers, largely as a result of either delayed or canceled diagnosis or a delay in preventative screening. One of the consequences is that there could be a tremendous increase in the number of cases of different cancers, and potentially even cancers that have been on the decrease due to therapeutic interventions. There may be potentially an increase in drug discovery funding for an oncology as a result of this, because there will be an increase in the number of patients that may present with later stage disease. So this represents a slightly different aspect of the oncology landscape, being able to treat cancers that perhaps previously were identified at an earlier stage. This makes the development of new anti-cancer agents even more important than it has been prior to the COVID-19 pandemic. So let's take a look now at some of the technology platforms that we have available and can apply to drug discovery. In particular here, we're looking at the PathHunter platform, which uses a proprietary enzyme fragment complementation technology to allow the assessment of a variety of different aspects of biology relevant to oncology drug discovery. For example, looking at aspects of signal transduction, protein-protein interactions, receptor translocation and trafficking, all within the context of a highly sensitive and robust platform. This is shown by the graphs on the right-hand side, where in the top plot you can see the impact of small molecule inhibitors of checkpoint receptors, and in the bottom plot, PD-1 signaling. From these examples, you can quite clearly see the high quality and high reproducibility of the data these assays provide. As I mentioned, we have platforms here that are available for evaluating a wide range of different aspects of biology. This can include looking at things like more classical targets, such as tyrosine kinases or other receptor biology, various aspects of immuno-oncology and nuclear hormones, both from profiling and screening modes, and aspects of looking at different pathways from both screening and profiling perspectives. Looking at kinases has long been a mainstay of oncology drug discovery, as kinases are one of the major target classes. We have a couple of programs with different platforms that can be used to evaluate kinase modulators. The KinomeScan platform makes use of an ability to measure binding of different agents to a variety of human kinases, more than 480 in fact in this particular platform. This can give information specifically on whether or not your modulator is binding. At the bottom, you can see highlights of Kinase Profiler, which is an enzymatic-based platform and a way of looking at the effects of agents directly on the modulation of kinase enzyme activity. So we have two different platforms that are complementary for characterizing how a particular agent is not only binding to a kinase, but actually modulating its activity. Again, it's just an example here of how Eurofins can help clients use different technology platforms that have been designed to evaluate, in this particular case, the kinase class of drugs. We also have a comprehensive and wide portfolio of ADME and toxicology solutions that can be employed to any drug discovery program in the oncology field, as well as, of course, in other therapeutic areas. These IND-enabling assays cover bioavailability, including the solubility and distribution of agents, evaluation of whether the agents are prone to be substrates of drug transporters. We can look at metabolic stability through classical CYP450 assays, as well as through metabolite identification and a variety of other aspects of drug metabolism. Our custom solutions can be applied collectively to evaluate whether a drug is suitable from a bioavailability or metabolism and excretion standpoint, as well as toxicology standpoints. When thinking about antibody drug discovery, we have a range of assays directed towards looking at aspects of antibodies that can contribute to the therapeutic efficacy. For example, looking at antibody-dependent cellular cytotoxicity, CDC, and ADCP, We see the example of trastuzumab, where we can demonstrate the ability of this agent to elicit ADCC. This reflects one of the contributing factors to its therapeutic efficacy, in addition to its ability to bind and block HER2. We can obviously look at these from a wide range of different perspectives. And in fact, many aspects of antibody drug discovery now are employing engineering to actually take advantage of these differential aspects of biology to improve and expand the therapeutic activity of any particular agent. We thus have a wide range of different assays and assay types that can be integrated across a drug discovery program for a smarter, more rational approach. This includes the use of functional or mechanistic assays to evaluate the effect of an agent directly at the target, use of cellular phenotypic platforms, for example, showing here the dual EGFR HER2 inhibitor lapatinib as an example in OncoPanel, ADCC assays as a way of evaluating additional therapeutic benefit from antibody therapeutics, as we've just seen on the previous slide, and our Biomap platform, a highly translationally relevant system with advanced analytics where we can evaluate the effect of anti-cancer agents, both in terms of the tumor microenvironment and in terms of a wider range of human tissue disease and biology models. And beyond this, ultimately, we can select the best model in which to evaluate agents from an in vivo perspective, collectively providing a smarter way to do drug discovery and for making better informed decisions earlier in the process for greater efficiency. Our OncoPanel cellular phenotypic platform can be used at all stages of oncology drug discovery. For example, from earlier stages where OncoPanel supports SIR screening or lead optimization, allowing one to rank order test agents against a few cell lines, all the way through to the evaluation of a candidate molecule against a much wider, broader range of cell lines representing diverse human tumor and tissue types. OncoPanel can be used even further by evaluating drug response data in the context of genomic data to identify predictive genomic biomarkers for both sensitivity and resistance. This can best help guide patient stratification for clinical testing and clinical evaluation of an agent, and can also potentially help identify areas of resistance, thus informing better combinations that might improve the activity of a particular agent. Ultimately, these decisions can help reduce the cost of clinical trials and improve the chances of success for patients. Again, this comes back to aspects of working with these platforms to reduce cost and improve efficiency, while also improving the chance of success in bringing much-needed agents to the clinic. We have a range of other phenotypic models available through our biomarker services and our Biomap platform that can help look in much greater detail at additional aspects of human tissue biology. For biologics, this ties in to how they may modulate the immune system, and from the safety aspect, by determining if biologics are immunogenic. Focusing for a second on the immuno part of the immuno-oncology drug discovery, we have a wide range of assays that can be employed to look either at modulation of the immune response or, from an immune safety perspective, evaluate whether agents have the potential for eliciting something like a cytokine storm response. This is obviously a very undesirable and potentially show-stopping aspect of biologic drug discovery. Furthermore, we can actually look at the effect of various agents directly on the immune system through assessment of immune cell phenotyping. Thus, we can identify what effects are going on in different immune cell types that may be important in targeting a particular agent's activity towards a particular immune cell. We can use the Biomap platform to more deeply evaluate the efficacy of immuno-oncology agents. And in this particular case, we're showing how we can do this with combinations by looking at the tumor microenvironment. In this particular example, we show how pembrolizumab, in combination with paclitaxel but not epacadostat, actually serves to restore relevant immune responses required for immuno-oncology efficacy. Using this platform, we can much more effectively and efficiently characterize how agents or combinations of agents are modulating the tumor microenvironment to provide greater therapeutic efficacy. One thing to remember is that cancer drugs are all ultimately fundamentally toxic, so ways of evaluating potential safety can also be paramount to improving the success of a drug. It's one thing to have an agent that's a good direct anti-cancer agent or that restores the immune function in the presence of a tumor cell, but one obviously doesn't want to have an agent that is cytotoxic in a broad sense or that can result in hyperactivation of the immune system. Our biomap platform is actually able to look at these kinds of toxicity signatures by evaluating the effects of drugs in a wide range of other different models of human tissue biology that are highly clinically relevant and extremely translational. As a result, we can identify potential toxicities earlier, and this comes back to the aspect of reducing risk and improving the efficiency of drug discovery programs. Identifying these potential issues earlier allows them to be resolved before, for example, selecting in vivo models or going into clinical testing. Finishing up this snapshot of a variety of assays that we have available to support oncology drug discovery programs, we also have a wide range of in vivo models available through our partner laboratory, Pharmacology Discovery Services. This includes conventional human tumor xenograft models and syngeneic murine models for evaluating immuno-oncology agents. These models can be used to evaluate a diverse and wide range of therapeutic modalities, including conventional small molecules as well as a wide range of biologics such as therapeutic antibodies, peptide molecules, and other novel approaches. We can also evaluate different strategies such as epigenetic modulation and PROTAC strategies, for example, to look at ways of targeting oncogenes by disrupting their stability. We can also look at a range of different cellular models, including CAR-T therapeutics, and these particular in vivo models indicated here. Collectively, then, we have a wide range of different technology capabilities and different areas of expertise, all of which we can bring together to support any drug discovery program in oncology. In fact, we can do this across the whole range of assays in this portfolio, including chemistry for small molecule drug discovery, both at the level of medicinal and synthetic chemistry, to bring to bear a complete drug discovery program. And in fact, if there is a need to do that, we can use our Discovery One capabilities to put together a critical path for a drug discovery program for clients. And on that note, I'd like to thank you very much for your time, and I will now invite our Scientific Director, Alistair King, to take any questions. Thank you.